Hi folks, so we're going to talk a little bit about we're going to get started on working on electron transport. The electron transport chain is a respiratory pathway. This is, the whole respiratory system is providing oxygen to it. And the ultimate purpose of this pathway is to funnel electrons onto oxygen. Oxygen is the ultimate electron acceptor here. It's the most, of course, we're trying to reduce oxygen, and oxygen, of course, wants electrons for itself. So we're really going to have to do some work to end up making oxygen. But the net result of this is that we form waters, right? So we get from a single oxygen molecule 2 waters, and we need 4 H pluses to do that, right? So we are going to try to channel or funnel electrons onto oxygen through this pathway, and we're going to generate waters, which is the second waste product of glucose metabolism, right? The other one was CO2, which we saw formed in the pentose phosphate pathway and in the Krebs cycle, among other places, right? And so the electron transport chain happens on the inner mitochondrial membrane. And if you remember your anatomy of a mitochondria, all these little folded cristae inside of the mitochondria are the folds of the inner membrane, which give it a lot more surface area packed into a smaller space. And essentially you can see it's kind of packed in there. And the inner mitochondrial membrane is actually heavily protein laden. There's only pretty much enough lipid there to keep it put together as a membrane, but mostly it's protein, right? It's majority protein. And that inner mitochondrial membrane is just packed full of transporters and accessory proteins and the electron transport chain. So what I'm going to start to draw here is kind of a blow-up version of what we see on the inner mitochondrial membrane, kind of highlighting the important features of the electron transport chain. Now, on the electron transport chain, we have 5 enzymes that are responsible. We have 5 what are called complexes. 1 through 4 are used to generate a protochemical gradient. Which is a higher H plus concentration on the intermembrane space, right? Remember, the intermembrane space is the spot between the inner and outer membranes. And we're going to use those to fuel number 5, which is our F0 F1 ATPase, to generate ATP. Okay. And so together, these guys typically are referred to as oxidative phosphorylations. Okay? So you'll see that a lot when we're talking about the electron transport chain, because that's really what's happening. We're doing a lot of oxidation of these reduced cofactors like NADH that we've been cashing in through Krebs cycle and glycolysis and all these other places. So we're oxidizing those cofactors, and then we're tying those to phosphorylation from ADP to ATP, right? And that's using complex 5. All 5 of these complexes are membrane proteins. They're associated with the mitochondrial inner membrane. So I'm going to draw these complexes kind of like this. Okay. So here's complex 1. Then there's complex 2. Remember that complex 2 is our succinate dehydrogenase that we saw in the Krebs cycle. So that's a member of the Krebs cycle. So Krebs is going to go through this cycle. And this is one member of that complex. Then we have complex 3 and complex 4. And finally, complex 5. And so we're going to talk about each one of these. Remember, out here is our intermembrane space. Inside is our matrix. And a big part of what these guys are doing is trying to generate that proton chemical gradient where we have lots of H plus out here and less H plus on the inside. Right. So we're going to see a number of these complexes actually working to generate that gradient. Okay. And an important thing to know right off the bat is that complexes 1, 3, and 4 pump 4 protons per every round of reaction that they do. Okay. Now 4 gets a little bit, 3 and 4 get a little bit complicated. Really 3 is the complicated one. But what you should understand is that for every time that we're finishing the electron transport chain and we finish an entire round in per pair of electrons, we're pumping 12 protons out. Okay, so for every pair of electrons, we're pumping protons. Now, remember where these are eventually landing is on oxygen. And that's delivered to our cells, of course, by hemoglobin and myoglobin and all those guys. We're going to be making those 2 waters, so we need 4 H pluses and O2. So we're going to be diluting or getting rid of the H pluses through attaching them to oxygen to make waters, but we're also going to be doing it through a number of different other factors as well as we pump it. So we're going to be depleting H plus on the matrix side, and we're going to be generating more H plus on the IMS. And that way, when the rubber hits the road, complex 5 is going to take in 10 protons, and it's going to be making 3 ATPs out of it. So we're going to take 3 ADPs, 3 phosphates, and make 3 ATPs out of that. For every 10 protons we pump back into the matrix, down the gradient, right? Remember that we have a lot more H pluses outside than inside. Bringing them back down the gradient is like passive transport, right? That's a diffusion kind of thing. But we're going to couple that diffusion back down the gradient to phosphorylation, right? So the idea here is that each of these complexes has a job, right? And they all do roughly the same thing, which is funnel electrons towards complex 4, which is where the oxygen stuff happens. And then all of them together kind of collaborate, except I guess not number 2, but all 1, 3, and 4 collaborate to bring protons outside so that 5 can make the phosphorylations happen. Now notice what's happening at number 2. Number 2 does not cross the membrane, so it cannot pump across the membrane. That's why we don't see that. And that also means that we get slightly different yields when we do different steps, right? And so typically what's considered to have happened in each of these steps is complex 1 is our NADH dehydrogenase. NADHs that we've generated through the Krebs cycle, or even glycolysis that are brought into the mitochondria, are going to be cashed back out. This is kind of like the cellular ATM, where we get our money out. I've been saying all along that NADHs are worth 3 ATP a piece. That's true, as long as you can cash them in, as long as the electron transport chain is working, right? I could have a whole bunch of money in my account, but if the ATM's not working, it doesn't matter, right? Because I can't actually trade my imaginary bits in a computer for actual cash, right? If it's not working. So we're going to be changing this back to NAD, right? But we're going to be funneling those electrons onto something called coenzyme Q, and we're going to be making QH2. This is essentially like taking a bunch of different kinds of currencies from around the world. Like if you got someone who has euro, somebody who has dollars, and then somebody who has, I don't know, renminbi or Chinese yuan, and trying to exchange those all for a common currency. So everyone comes to Paris, you exchange those for euros. Everyone goes to Russia, you go to rubles, right? The same idea. We're going to exchange all these different currencies, these NADHs that we've been building up, the succinate from the Krebs cycle, and a whole bunch of other kinds of things into a common currency, which is QH2. Right? Now, coenzyme Q is a cool little carrier because it has a lot of properties that make it very useful. Okay, so this is what we call ubiquinone. The ubiquin thing means because it's kind of ubiquitous. It's pretty much in all tissues that are respiring. And so it's OCH3. But ubiquinone has a long nonpolar tail. And we call it CoQ. But this thing can be between 6 and 10 units long as a repeating polymer. So essentially it's this long branch chain where we end up with, you know, a series of, essentially it could be 10 units long. So if you've ever seen CoQ10 in the stores, this essentially is ubiquinone, and it's thought, you know, as a supplement. This is pretty neat because, you know, what if you run short on this? Then that'd be a real problem. And sure, of course it would, but we can make it just fine ourselves, and we get it from our diet because it is ubiquitous, right? Now the cool thing about this is that it can accept 1 or 2 electron at a time, and it forms what's called a semiquinone intermediate. So 1 electron and a hydrogen. What we do is we form a radical semiquinone, which is stable, and so this guy can hold on to 1 electron at a time, so it's a radical. Or we can pick up a second one and form what's called QH2, or ubiquinol. Okay. This is ubiquinol. This is CoQH2. Sometimes I'll call it just QH2. Whereas this guy I'll call Q, and then I'll call this guy QH dot to show that it's a radical. Okay? And we'll see that coming up here as we start to talk about what's going on with Q. But the reason that we have this particular cofactor instead of something like soluble NADH that we saw earlier is that, you know, NADH is cool because it can float around in 3 dimensions, and if you're a cytoplasmic protein, NADH is something it's good to give off because it floats away from you, right? You have to get rid of it somehow. Why not just toss it out the window, as it were, toss it out of your active site, right? But unfortunately, it also introduces a lot of complications when it comes to efficiency of finding something, right? So if I was to go and say, I want to go play hide and seek with you in the Sears Tower of Chicago. Go. I would go hide, and I'll be on the 88th floor, and you'd start systematically searching floor 1, 2, 3, all the way up. You might never find me, at least in a reasonable amount of time, on the Sears Tower, because that place is enormous, right? It's huge. There's multiple floors to check. It'd be fun, but you wouldn't be very efficient at finding me. Now, if I said, I'm going to be on the 3rd floor of the Sears Tower, go, you would probably find me somewhere because I've narrowed it down for you. And the same kind of thing happens for coenzyme Q. Taking it from 3 dimensions, going on the 3rd dimension, that height dimension for the Sears Tower, down to a single floor, makes it much more efficient for you to find me, right? And so for enzymes like complex 3, which need to find QH2, it's really makes them much more efficient than having to try to wait for them to diffuse to you in 3 dimensions. It only diffuses along this one sheet, and that makes it a lot easier to find you, right? And so QH2 is really important as an exchange rate because essentially, again, it changes all these different cofactors like NADH or succinate. Eventually, we're going to talk about fatty acids. Into a common currency, right? So just like you might exchange your euros or your dollars or your yuan or your renminbi or whatever into a common currency when you go travel, right? You might change them into rubles if you go to Russia or Canadian dollars if you go to Canada. Just so you have a common currency from all these disparate different kinds of money, it makes it a lot more efficient to run, right? So having QH2 as a common currency in the mitochondria is very useful. Okay, so I've kind of already talked about what happens here with complex 2. Succinate goes to fumarate. But also, those electrons are going to get cycled onto QH2, right? They're going to do the same thing. So both complex 1 and complex 2 do the same thing. But the difference is that complex 1 pumps protons when it does it, right? It does not waste the chance to pump protons, whereas 2 just is not able to do that, right? And so both of these guys kind of have redundant roles in filling QH2 in the membrane for the next complex 3 to use, right? So QH2 in complex 3 is recycled back to Q, right? And it pumps protons in so doing. But those electrons that come off are largely funneled onto cytochrome C, which is a intermembrane space soluble protein that is stuck in the intermembrane space and essentially is a shuttle between complexes 3 and 4. But cytochrome C can only accept one electron only. It's like a heme group in hemoglobin. It can only go from iron 2 plus to iron 3 plus. And that's really what's happening here. It goes iron 3 plus to 2 plus as it picks up an electron. And then it carries those electrons over to complex 4 to drop them off. Okay. Of course, again, we're pumping 4 protons every time we do a pair of electrons. And so we're going to be generating 2 cytochrome Cs from every pair because each one can only hold 1. And then we're going to need a total of 4 cytochrome Cs to channel the electrons down here onto oxygen. Right? And if you can count your oxidation numbers here, you'd believe me that I have to have 4 hydrogens or 4 electrons to attach 2 hydrogens onto oxygen. Right? We have to break a double bond and the single bond to attach those hydrogens. Right? So we're going to need 4 cytochrome Cs, and those go from cytochrome C 2 plus to 3 plus. And so I'm going to name these guys right now. Of course, this is succinate dehydrogenase. We've seen that before, complex 2 in Krebs cycle. This guy is ubiquinol, or QH2. I'll just call it QH2. Ubiquinol, cytochrome C reductase. And then complex 4 is cytochrome C oxidase. And that's really where the business happens. And of course, we've talked about complex 5. That's our not F1 ATPase, right? So kind of a big picture of this whole story. Complexes 1, 2, 3, 4 are involved in channeling electrons onto oxygen in some way. 1, 3, and 4 are involved in pumping protons out. Those proton pumping steps are used to bring hydrogens back in and make ATPs out of them. We're going to talk about accessory proteins for the electron transport when we get a little deeper. But ultimately, this is kind of the big picture of what's going on in the electron transport chain. So don't lose the forest for the trees. We're going to talk about each complex individually, how they work. A couple of important features here are, what they work on. The NADH is reduced, the succinate is oxidized or whatever. The NADH is oxidized to NAD. Succinate is oxidized to fumarate. The QH2 is oxidized to Q. That's the oxidative part of oxidative phosphorylation, right? All of these cofactors are being oxidized in order to reduce our ultimate acceptor, which is oxygen, right? The only thing that gets reduced here is oxygen. That's the purpose of this whole pathway, is to drop it onto oxygen. It's also why we need oxygen to be alive, right? It's the ultimate acceptor of all of the electrons that we've been collecting all this time. We've been catching them all, and now we got to cash them in, right? Just like maybe you had a Pokemon card collection as a kid. Many of those things are worth money now, right? So you're going to go cash them in. You've been holding on to them all this time. Now it's going to be your big payday, right? So as we go into the next few videos, complex 1, 2, 3, 4, and 5, keep in mind this big picture, right? They all kind of work together to generate one common output. Thanks.